big introduction. Anyway. All right, so anyway, yes, thank you guys for being here. My reason for doing this is I started working for the Department of Agriculture not too long ago, back in May, and so I have all these things, these resources available, so I'll be more like a courier of the information rather than I actually know something. It's just these are things I've gathered, and I found to be interesting and inspiring with regard to what is the future of citrus for dooryard growers, and then we'll get some insights into what the industry is either looking forward to or facing still. And what I try to do for us is to lean more into organic or interventions that are not necessarily utilizing chemicals and things. But I will also mention what their protocols are out in the field. My emphasis will be on greening, the HLB, which is the sort of nemesis of whether you're dooryard or you're commercial, and it's really affected the commercial industry. Up until recently, they've just been dealing with steady decline, and then they have to take things down and then do resets and things like that. But they're slowly rounding the bend, and that's, again, some of what the good news that I hope will come out of this little talk. Little background on the HLB, or Huanglong Bing greening. It's a bacteria, but it's kind of a not a plasmid, but it's a protobacteria, so it's not putting a Hoyle bacteria, as I understand it. The Asian psyllid, the green Asian psyllid, is the vector for it, and that came to the U.S. in 1998. And then not long after that, in 2005 in Miami-Dade, they started detecting HLB in greening, and then by 2017, it was statewide. It was documented and vouched in every citrus area in every county that was growing citrus. And so there's not much they can do about the insect right now, but there are means to some control by way of bio agents, I guess, other insects, allies. I'll touch on that in the talk. And I will start sort of at the top and work our way down into dooryard and just share some things that are going on with the guys in academia that are working in partnership with ag and working with the industry itself. One of the things that they're doing is they're seeking rootstocks that are going to be able to tolerate what HLB does to the root system. So there have been some breakthroughs in there. There's something called Super Sour 3 that a guy I know from the USDA is all excited about. There's something called the Donaldson Zion, which is from a Donaldson Groves up in Central Florida somewhere, maybe, I'm not sure, Lake County or somewhere in that area. And they found a batch of trees in this one grove that were resistant, relatively, to the effects of HLB. So they took them into the lab and they were checking it out, and now they're issuing Zions or Budwood for that and giving it to the commercial growers. I say that because basically it's a trickle down. If they're getting success in commercial, then we'll see it in dooryard probably within a few years. And so that's something to be optimistic about. And the one thing that I really thought might be practical for us for growing is that they're actually using a tenting technique as one means to really knock down most of the bacteria that comes, into the tree from the psyllid. And what they have been doing in what I could see in the literature is that it has to be a minimum of 95 degrees, and depending on the article that I read, sometimes it was 10 hours that you would need sustained, minimum of 95, up to 130. And so I'm thinking, for things that are not involving nasty chemical agents or weird stuff that I'll get into later, this is a nice, easy, accessible kind of thing. You just throw a tarp over your tree, keep it airtight. The big guys are using things like steam, and they're doing hoop houses all the way up and down roads, and it's like $1,000 to $1,500 per acre because you can just repeatedly move them. I could not find how long this works for. So if another batch of psyllids come in, you probably have to repeat. I could not find anywhere to say this is effective for 18 months or a year or whatever. So that I could not come up with. But Oh, I remember what I was stumbling on earlier. I have a YouTube video that I'll put into the Facebook group, and it's an unlisted video. Down in the description field will be links to everything I'm talking about, so that if you want to dig deeper and look into the informal articles from IFAS or into the deep literature where they have all the technical stuff, that's all down there if you guys are interested. So I was thinking for us, for what I found is that scenting seems to be a really good option. The efficacy is pretty high when it comes to knocking down the amount of the bad things that happen with the bacteria within the phloem and within the juice of the tree itself.
Is there a certain age of the tree when you can do that? And is it just like one day that you do 10 hours and then you're good for like a certain amount of time?
No, it's a longer period it's a longer duration to do it. One says 96 hours, which is 3 days, up to 130, and then the other says 10 hours to a day. And I think the cycle for the 10-hour a day was a week or 10-day interval. So it's repeated, but that ensures that you'll have a higher rate for doing that. And again, there was no real indication for having to go back and do it again, but that they were getting reasonable results, and that they were willing to do it in commercial. That struck me as being a kind of an easy out for us. You know, when we're growing, keep your trees pruned so you can access the fruit, and then just get whatever it is that you think is economical tarping means, or there's those the greenhouse companies that have like a malleable plastic that's like a drape. I mean, I didn't go that far in my research to find out if there's something like a kit that you can buy. Probably not, because this is still pardon.
I'm so sorry. Aldi's have. I saw on Facebook. Aldi's has these like, it's like a metal structure. I think it's $20. It's like a metal structure.
Okay.
I think it gets cool, but it's
right.
Like a metal structure that you put around it and you put like a bag over it. And
yeah.
For an individual.
Exactly. Yeah. Some kind of improvised system like that would be awesome, I would think. And so for me, I guess the control is that you want to have your thermometers in there, and they also said to meter to check temperatures high and low. So go like a foot off the ground, and then go maybe up in the upper canopy with two different thermometers to just try to get a mean for yourself and figure out where you are with temperatures and things like that. So that's one option. So now I'll break away from what's practical for us, and I'll talk about what the industry is doing that may, again, downstream to us. And one of them was it's a transgenic process that they're working with. And it's kind of adjacent to GMO in that there's a subfamily called Aurantoideae in citrus, or in Rutaceae, and then Aurantoideae is what citrus is in. And then within that, there's a cousin called Citrus australasica, which is the finger lime. They're finding that the finger lime is really resistant to greening, and so they're doing this transgenic introduction into, say, a Valencia or something like that. And they're finding stability in being able to sort of bring in whatever that might be RNA pieces or full genes, I'm not sure. But anyway, they're meshing this together, and it's nice because like when they were talking about GMO for this kind of thing, they were actually taking spinach genes and putting those into citrus trees because spinach has a high antibiotic, has a means to deal with things in a greater capacity. What they were finding with the transgenic though is that citrus tissue culture is recalcitrant, is what they were calling, which means obnoxious. It doesn't want to do what they're asking it to do. So these recalcitrant, no matter where they were getting these explant cuts, whether it was a node off of like an emerging budding part of the tree, or if it was off the cork, they were trying different parts of the tree to get less recalcitrant reactions from it. They finally zeroed in on it. So this is kind of weird, techie, geeky stuff, but it's like that's what they're doing to try to bring in not quite GMO, but something adjacent to it, right, that will help both commercially, but I also think trickling down to where we are, where we'll get trees down at Nelson's or wherever, Peterson's, that will be mostly insurable against, and there's variation within HLB. There are, I can cheat and get the name. It's called Candidatus Bacter or Liberbacter, and there's like not species, but, where there's other variants of it. So they have to contend with that too, that there could be variation. You get something that might fend off one subspecies or whatever you want to call it, and then, so they're still dealing with that. But so the transgenic thing is a possibility. They're even using gold and tungsten nanoparticles to try to bombard the, it's not necessarily the cell, but like the tissue, to get it to inculcate the new strands of RNA or DNA or whatever it is that they did to take within the tree without having to go the clone route or the tissue culture route. So I just thought that might be interesting if you like that kind of nitty gritty. And they also use something called an agrobacterium, which is a carrier. Like sometimes they use carrier viruses to do CRISPR or RNA and stuff like that. So they actually use bacteria to transfer that over. So that's too geeky. I apologize. Now, going back to biocontrol for a minute, there are hoverflies that are vegetarians when they're adults, but the instars and the maggots are actually carnivores or bugivores, and they love the psyllids. And so hoverflies are one of the biocontrols that they're working on. I don't know how practical that is for dooryard, because this hoverfly probably needs critical mass. Yeah, it needs a lot of sustenance. It needs a lot of its own, this kind of ecosystem to sustain itself. But the good news is that these guys also work on a lot of things in the nightshade family. So there's like tomato psyllids, there's potatoes, there's all, who knew there were all these psyllids? There's psyllids for everything in the nightshade family. So if we're growing eggplants and tomatoes and so forth, we might actually have a host community that will help sustain the hoverflies. Mm-hmm. And on that note, there's guys down at Heathcote for the USDA, part of their adjunct to the Pecos Road. They're working on developing allied plants as a kind of downstream or cascading support system for these hoverflies, so that, they'll grow allied plants that attract certain, either the hoverfly cycle or other insects that will help with continuing the population of the hoverfly. And they're actually going to talk citrus growers into growing these type of these plants that will support ally, predation cycles or whatever. So that's another technique that I think we dooryard growers can use to have nice-looking plants, flowering plants. I can get that information if anybody's interested. They haven't published very much at it. The guy's name is Joe Pat. He's in the description field as well. And he and Larry and Elena down there are doing some really neat work. And again, it's with not having to use chemicals in mind, that because up until now there's nothing you can spray. They've tried spraying things onto the trees to the one thing that had some effect was kaolin clay, actually. And they put that on there because the psyllid, in its infrared vision or whatever, can't see through the kaolin clay, which is sort of a rusty red, it turns the trees kind of brick red for a while, and that just throws the psyllids off. And that, not only that, but it helps with UV light, I guess, during the peak of summer to help keep down stress. There was one other beneficial thing for that, but kaolin, and that's something we could actually try ourselves, because you can get kaolin clay, theoretically, you can order it, and I don't know how you make a slurry out of it that you can actually distribute in a mist. They have those big blast sprayers in the grows, but anyway, if you're motivated. Is it a normal sprayer? I don't think so, because it is again, you could make it so you could make it less viscous-ish or less.
I think people use it in California just for the heat, right?
Yeah, to keep it.
And they just use normal sprayers.
Oh, really? Okay, good. So you can water it down enough to. Okay, cool. Thank you.
It doesn't clog.
Oh, good. I thought, I was wondering that, because I imagined that the big guys were using those big blast sprayers and you just 50 million psi pushing it out. Yeah. So kaolin's a nice thing for us. Now, the one thing that they are using out there, which I think is controversial, but again, I've tried to keep everything as organic as possible in this talk, is they're actually using something called oxytetracycline hydrochloride, which is a lot like, what's the other one that they're using in the horticulture for the lethal bronzing? There's another one of those antibiotics, but anyway, they're using this oxytetracycline in citrus trees as a injection, a systemic, and it's had amazing results. I've actually been out in properties where about 6 weeks after they've done the injections, the trees are, kind of just reviving. So, but there's the antibiotic thing, I mean, this is, orthotricycline is what they're using in horticulture for lethal bronzing. But anyway, it is over the counter. It is available as a treatment protocol. But again, are you averse to antibiotics? I don't think the biotransfer or accumulation is an issue. I don't know. And I meant to ask my supervisor, like, what's the waiting period between when you inject and when you can eat or harvest the fruit? I didn't find that out. If anybody's curious, I can ask somebody in my division or wherever I can get that information from. I'll be happy to do that. So, yeah.
Is that a permanent treatment per tree, or is that something you have to?
I think it has to be updated, yeah, because you're constantly going to have new psyllids coming in, and that thing will wear out the efficacy. So I would intuit that that's something that would have to be done. And again, I don't know the cycles, like how long they do it. But apparently, when they do the treatment, it really turns things around and has some staying power to it. So let's see.
And how do you inoculate a tree?
Inoculate with.
If you're putting the antibiotic, you're just putting like a.
They actually have IV bags, if you can believe it.
This
I've not
is
seen
what
it.
I wanted to know.
Yeah, they hang, just like in the hospital, they hang the little bags out on the branches. And my one supervisor said he's been out there on cold mornings before, and when that sun starts to hit and the metabolism of the tree starts to activate, they said the bags just go like the tree, the vacuum or the pressure of everything moving up just draws it right out of the bag. So, but typically they have guys that go in, and bless their hearts, because they go right to the base where the rootstock meets the mesoxion in the clone, and they go right, I think, at that junction, and they pop it in there. So that, these guys, I don't know, maybe they have longer sticks if they do the shot, but you can see like about a 4 or 5 millimeter hole in all the trunks. You'll know which properties have been done. And sometimes we look just for funsies to see if that, if that property's been treated. But again, I don't know what happens to us when we eat that fruit. So I'm agnostic about it, but it's what's going on out there, and if people are not terribly averse to using it, then it's something that can be acquired and purchased. To speak to, let me see before I get. Oh, and there's a name for that hoverfly. It's called the oblique streak tail. And there's another ally insect that we have. It's called Tamarisia, and it's a parasitic wasp. And I actually saw a guy in Merritt Island about 2 weeks ago, and he had a little jar full of them, a little plastic vial that he got from either IFAS or the USDA. And they just go and turn them out in their fields. Now, again, for us, I don't know if Tamarisia is going to enjoy our yards as much as hoverflies might, because, again, they would feed on other psyllids. But maybe if it's a parasitic wasp of psyllids, that there are allied species that we grow. I was mentioning for the hoverflies that the solanaceous vegetables like eggplants, tomatoes, potatoes, all that kind of stuff, they all have their own nasty psyllids. And so if we are gardening, if we only have 1 or 2 citrus trees, how are we going to support these poor little hoverflies in their life cycle? So that if we have other allied plants, that they can. I'm not sure if you were out of the room for that part.
I was.
Okay, yeah. So the hoverfly is an option, and then the parasitic wasp is an option. They can be had from either. I can get more deets if people want to know where you can get them from. I can probably check with my supervisors and stuff, and see where they get those from. Let's see. Trying to figure out if there's anything. Let me move from greening to a few other things, and then I'll ask if you guys have any questions, and we're probably getting to that point. Okay. The other things there's a lot of stuff, and I have flashcards over here that deal with some of the more exotic diseases like greasy spot, melanose, bacterial spot. There's something called lebic. It's a mealybug, and it's nasty looking, and it's not pernicious. It won't destroy the tree, but it's just, it's gross. It kind of looks like bird manure or something, guano. It's just a nasty. We had pink mealybugs in hibiscus for a while. Do you guys remember that? And they would really knot up the hibiscus, and it looked really gnarly. Well, that's kind of what they're doing to the citrus trees. That's something that can be treated with simple horticultural oil. So if you do have a citrus tree, you go out and you see this, and I've got a flashcard over there, this hideous popcorny, black and white, sooty, nasty, fungusy looking thing, any basic neem or horticultural oil will knock them down. So that's a nice one that doesn't require heavy intervention. On to canker, which, bless you, when canker first came out, it was interesting, and I don't remember the dates, but the canker came and went, we conquered it, it left, and it came back, and da-da-da-da. But now it's kind of ubiquitous. And the one thing that they're using out in the fields for that is copper spray. So it's kind of nice in a way, but then when you apply over and over again, you get the accumulation in the soil is kind of nasty. So, but for us, for dooryard, if you do ever see canker on your leaves, and there are flashcards with pictures of it over there, that's the effective treatment to knock it back. It's also a bacteria. I always thought it was a fungus, but when I started working for them, it's another fungal or a bacterial issue. But yeah, that fortunately, again, that's something that can be treated without using really nasty, drastic chemicals. So I'll stop there, and if anybody has any questions, I don't know if I covered everything on my sheet, but yeah, if anybody has questions. Oh, and on the YouTube video, I do have a thing from, I think it's IFAS, and it talks about organic options. There are very few, but there are some practical steps that you can follow to make sure your trees as healthy as they can be, given that you want to keep it OMRI sacred or keep it all up to snuff.
I heard early on that citrus was doing better under oak canopies or something. They're researching something with oak leaf extracts or something like that.
Okay.
Have you heard anything about that? Do you know if there was anything?
I've not, and what I've noticed that trees that are growing, there was one up in Scottsmore, up on the Volusia line, and the guy had a little postage stamp block, and he had some live oaks shading out some of the, so the cascading sooty mold was an issue for him, because, oaks just constantly rain sugars down, and so anything that's underneath it. But I've not heard, so is there something that's going on underground, or?
Okay. I didn't follow it, and I knew it at some point. I heard that research is happening in it, but I don't really know that much about it.
Okay.
If you would.
Okay. Again, the only thing I've ever noticed is that when they are shaded out, that they can have that issue that you'll get with palms or anything else that's growing underneath an oak tree. One other interesting trivial note is that the guys that do cloning and selection had come up, in 2018, they came up with 14,000 new potential budwood or Zions, and with 7,000 more on the way. And I'm thinking, how do you even keep track of all? I mean, I know they have databases and stuff like that, but it's like to rigorously test these things to see, just blows my mind. And they also have a couple places in Florida, kind of like the seed sanctuaries and the places where they store things for safety. They have places in Florida where they keep both rootstock and Zion, budwood type stuff on hand that's hermetically sealed from the world, and they're trying to keep things there in case something weird happens and another disease comes along. And I went to a talk in May. There was a mycologist or a mushroom guy over in Winter Haven, and one of the people that introduced him said she was a field supervisor, and she said some ridiculous amount of new pathogens and vectors are coming into Florida every month. I mean, it was just nuts. Just, with commerce and all the things that we bring in from importation. So it's amazing. But anyway, I'll hush for a minute. Anybody had a question?
So a couple of years ago, I wrote some grants for some of the bigger groves to the state of Florida that paid for things like bagging and rootstock.
Mm-hmm.
I haven't had a chance to follow up, but the bagging was like that filmy type of stuff. Do you know if there's been any success with those? I mean, they were handing them out with candy, and I guess it probably didn't hurt to try them, but
it should be effective for greening to keep the psyllids out. Unfortunately, they call them craft blocks, and so they're the resets that are underneath the little tents of the very fine mesh, and but we found canker has gotten in there. So canker inoculums, whatever's going on, is so tiny it must get through this really, really fine mesh. And Val had a great point yesterday that we can include in this, and that is she intends to grow citrus in containers in a fine mesh screen environment on her porch, and so that is an option that
The bags.
Right, but nonetheless, it'll keep the adult psyllids. Yeah, so that's a great idea too, is to do that. I might even, because we're gonna fix our one breezeway, and maybe we can mesh it up and keep it safe that way. So, but yes, I would say to greening that it is probably effective, because they're still using them. And
Right, yeah, I see them.
yeah, there's a lot, and it's wild to go out and you'll see a block with like 1,500 trees, and they're all under these little canopies, little things. So, but I would say, yeah, but again, some things get in that they can't control. Any other questions?
The scion that you said that they found at that property or whatever.
Yes.
What type of citrus is it? I mean, is
It was an orange, and if it was a Hamlin or I can't remember what it was, but it was a real like ubiquitous trade type orange, probably like a juice orange, not like a walk into. Do we even have those Kennedys or anything in like Peterson's has stuff still. But yeah, it's not one of the things you walk into the showroom and see the glorious fruit kind of thing.
but they're they're using that not so much for well, I'm sure they're doing both, but for like genetics research purposes, it's more for this is an actual practicable scion that we can distribute and and use in groves kind of thing.
My intuition is that when they were distributing cloned specimens, is that they went with that transgenic route, perhaps, or they just went with straight tissue culture, but they may have tried to stabilize it, because obviously, just because 2 or 3 trees may not be showing it, there's really not much proof there that it's going to carry out. And there's always genetic drift in what I understand from cloning, and not so much tissue culture, but like raw cloning from just scions, that you get drift if they clone a clone kind of thing. So you get some drift out of that. So I'm not sure how they're keeping everything straight with that, but I would imagine it's just a tissue culture process that they've managed to be good at, to get.
But they're not. It's not as much like, hey, here's some that are resistant. Let's figure out why and try to use.
I'm sure they're trying to reverse engineer.
But it's mainly like here's let's make a bunch of these so people can have groves that won't have greening issues.
Yeah, I wouldn't be surprised if they, and I can find out, I can dig, but they may be sequencing things and saying, okay, well, what's different from this tree versus the other trees? Maybe through sequencing they can find out. I don't know if just reading, gene pairs, gene lines and stuff is going to tell them that, but it was a fluke. I mean, it was just these 2 or 3 trees that were robust, in the light of a otherwise devastated spot. So I don't know. Could be something in the background, because I don't know what is that X Paradisi or whatever those oranges are. Maybe there's something in the background there that has that potential, and it just came out in those trees. So I'm not sure, because obviously those trees were cloned, so everything should be really stable. So I'm not sure. But I can find out more, and next time I see you, if I find anything else, blast you with some Cliff Clavin trivia. It's a little known fact there. But yeah, I'll see what I can do, because I'm curious myself. And that led me to think about something else that just slipped out of my head. Oh, there was an amazing documentary. If you guys want to go on a deep dive on the history of citrus, going into before human and ethnobotanical, the period of our using citrus, there's a documentary that I'll dig up, and I can put it on the Facebook page. That's absolutely astounding. This kid did his homework. He's I think he's an anthropologist. So he just put anthropology stuff into citrus and showed how maybe 2 or 3 disparate species drifted out, radiated out naturally, and then when we took over, and then what we did. It's like an hour-long talk, and lots of neat graphics and stuff. So anyway, good one if you're into citrus. Other questions? EJ or Val? Or no pressure. Just yes.
I heard it's made its way over to California.
The green?
Yeah.
You would know more than me, yeah. She's from San Diego.
I think last year they finally found it in a commercial grove, but up till then it was just in a handful of trees and backyards.
Mm.
So they're slowly quarantining little sections here and there. But California isn't as good of an environment because of, well, it's cold, I guess.
Right.
Maybe the aridity too, like can the psyllids, I know they're originally from probably Western Asia, from China. So I wonder if the aridity might also help.
The psyllids are slowly making their way to Northern California too, but that's even harder. for them to survive.
Colder, probably.
Yeah.
That's too bad, you guys, or they, or you. You're one of ours now, you're not a California person, you're Florida people. So, yeah, if there's any other questions, I think that was everything I had as far as neat stuff that I found. But, yeah, again, on that description field of that YouTube video that I'll put up as soon as I get home, lots of neat stuff in there. And the articles some of it's really mind-numbing technical and others is really accessible, so. Anyway, so it's how much you want to dive down that citrus rabbit hole. It's up to you.
I deal with that.
I was looking at, okay, then you might enjoy it. I was looking at the peptide thing and how they were, again, blasting them with little nanoparticles of gold and tungsten to get the DNA to take it up or whatever was going on. I was just like, wow, this is pretty wild. So, anyway.
Thank you so
Yeah.
much for coming.
Yeah.
You know,
Yeah, thank you guys for
it's
coming.
about quality and we're the best.
Yeah, you guys are the cat's meow. And absolutely.
So we've got the graphics here if you want to come and look at some of them. And then also with regard to the plant, you guys do your own thing on that, since there's only 3 plants. Figure it out. I don't think we.
Thumb wrestle.
Yeah, you. But yeah.
And correct me if I'm wrong, I've always been told, it's when it attacks the plant, the reason the plant starts to decline is because it cannot draw nutrients.
Compromises the roots themselves.
Okay.
So, and there's one other critter I can talk about too, real quick while everybody's milling around. But yeah, it really messes up the root system, so then uptake becomes difficult. There is another critter called Diaprepes brevicus or something, and I think it's called the root weevil. And we may or may not deal with this in dooryard citrus, but it's really the bane of the root systems when we talk about that. Reminded me. I don't think I have anything on the Diaprepes over there, but those things are obnoxious too. They'll wipe a grove out if you don't get the intervention right away. And I'm not sure, they probably have to use pretty nasty chemicals for that, because they got to get into the root system, because that's where it's either the instar stage or some early stage of the bug is down there, and then they gravitate up, and then they destroy the top. So it's like there's insult to injury in these life cycles of these guys. So